And now we're going to move on to Thais de Oliveira from the University of Sao Paulo, Brazil.
Hello all, I'm here to talk about Plasmodium vivax and the main malaria hotspot of Brazil and what population genomics can tell us. So first, I'd like to thank all the organizers that made this symposium happen, even though we have these difficult times in pandemics. So in the next slide, I'm going to talk about a little bit about malaria vivax. Plasmodium vivax is the geographically most widespread malaria, human malaria and parasites, and cause significant morbidity in Americas, Middle East, Asia, and Western Pacific. And it appears to be more resilient than Plasmodium falciparum to the currently control and elimination strategies. In the next slide, we are seeing that some general data about this disease, and we have more than 40 million worldwide cases, and from them 700,000 cases just in the Americas. And from this case, we have 16% of all these cases in America, just in Brazil, from Plasmodium vivax. And next slide. So although the countrywide incidence of malaria has dramatically decreased in the last decades in Brazil, we have this residual transmission in the Amazon rainforest that have 99% of all the cases in Brazil. But in the next slide, thanks, 18% of all malaria cases in Brazil occur in just one region in the Northwest. What is curious, because we have just 4 cities in this region. And the main city that we have there is what we consider the main malaria hotspots, is the name of Manso Lima, that we are going to call ML. In the next slide, I'm showing you a bird eye view from the city. As you can see, the city is inside the Amazon rainforest, but agricultural and mining activities end up resulting in this intense and continuous deforestation, as you can see around the city. And what this leads us to a lot of migration in the region itself, and the regions that are closer on and regions that are not so close to this one. In the next slide, I'm showing you our main question about this work is how is the transmission in the main malaria hotspot of Brazil? Could be like possibly just local transmission, could be the total opposite, such as the infections are dependent of this continued introduction of new parasites, or could be something in the middle when you have a local transmission and you have the introduction of new parasites from other places. In the next slide, I'm going to show you how we try to answer this question. So we had blood samples from the patients, and we take some socioeconomic and GPS location from these patients. After, in the next slide, we did some diagnosis using qPCR and microscopy to make sure that we just have Plasmodium vivax infections and not co-infections with other species such as Plasmodium falciparum. And next slide, we end up with 56 patient-derived whole genome sequences that we did the bioinformatic analysis in the next slide. We did all this SNP call because we want to use SNPs as markers, just monoclonal samples, and we add some samples from other cities. One is closer and one is not so close to this one, to compare and see how the transmission is working. On the next slide, it's our whole data set. So we are using like 3 cities, samples from the ML population, the main malaria hotspot, and the 2 other ones. One is Cruzeiro do Sul, that we are going to call CS, and this city has like a lot of cases of malaria too, and in the last years have been like epidemic case in this region too. And we have one city in the that we are going to call AC, Acrelândia. The city has been decreasing the number of malaria in the last years, and we have more than 7,000 SNPs in the total. First, we are going to just look for ML city, and then we are going to compare this city with the other 2. So in the next slide, we have the first result that we have is just a number for you, but this number is the main nucleotide diversity from this population, and this number is like as diverse as predominant outcrossing in Plasmodium falciparum populations in Senegal, or even in populations in Southeast Asia, Plasmodium vivax, and where you have like high transmission. And in this area, we don't have like this high transmission. But it's not surprised that this population has this high diversity because we saw in other studies from Plasmodium vivax in the Americas that we always saw this high diversity. What is surprising that we have this high diversity in the last continent that was colonized by humans, what is not supposed to be. In the next slide, we continue to look at the nucleotide diversity. So here I'm showing the graph that you have the nucleotide diversity within this population by 1 kb window. And as you can see, you have the distribution of the pi values that are negatively skewed, and you have this heavy left tail closer to the 0, and correspond to the pairwise comparisons within pairs with highly related parasites. Here, with some other results, we suggested that we have this relatively high population level genetic diversity in ML that could be result from the co-circulation of multiple relative distance inbred lineages. But to make sure that what we are looking from this population, and we are seeing this population that have this co-circulation of multiple parasites, we go for the next slide and look a little bit more about LD, the linkage disequilibrium as well, seeing if this population is under pamixi or not. So in the left graph, we are seeing the distribution of the genetic distance between each parasite in the nearest and its nearest neighbor. And in the red line, we are seeing the expected under random assortment in a pamitic population. So as you can see, we have this half-tailed of pairwise genetic distance in ML population that clearly deviates from what we are seeing in the red line, what we expect in pamixi. And this is the same that we are seeing in the right graph. This graph shows us the VDV ratio. So what is VDV ratio? VD is the variance of the distribution of the empirical pairwise genetic distance between local parasites. It's almost the same that we are seeing the right, but at the same time it's divided by what we expect under random association with allele in pamitic populations. So we expected that if this ratio is closer to 1 or equal to 1, this population is in pamixi. What is not happening here? Here, the VD exceeds largely VE in all the simulations with random subsets that we did. And these finds are constant with a highly significant genome-wide LD. And it's a suggestive of this extensive inbreeding in the parasite population. So if you take it together, these results with nucleotide diversity that I showed in the slide before, we have that ML population is fragmented into discrete lineages of closely related parasites. It's not what you're supposed to see when you have like a small city that you think that the parasites can recombine without any barriers. So this could be like a reminiscent of wallow defect, that you have separate lineages living in sympatry but unable to recombine. In the next slide, we are going deep in this population and see the identified descent, and using, in this case, you're seeing a network as you have each dot as a sample in ML population, and it's colored by the time they are collected. As you can see, you have like a larger cluster in the bottom, and that represents 16 samples that comprise closely related parasites circulating between 2014 to 2016, most of the points that we had the collection samples. So these finds are constant with this local propagation of highly inbred vivax lineage spanning 26 months. In the next slide, after we did all this deep analyze looking at the ML population and what the transmission are in this population, we want to try and understand the transmission between these populations that we have ML city, the main malaria hotspot, and the other two. As you can see, ML is in purple, CS is in pink. CS is the one that is closer, it's just like 40 kilometers away. And you have CAC, that is the one that is farthest and has been decreasing the number of malaria in this region. And as you can see, we have this cluster again with most of the ML samples, and the other, but as you're seeing again, AC and the farthest way is in all of the other clusters too. So we found significant ancestry between parasite at a large geographic distance, what is not expected, and with it's constant with extensive gene flow between these regional vivax populations in the same state. So in the next slide, please. So overall, we conclude that we have a highly inbred vivax lineage spread over time that we saw on IBD analyze and even in the LD analyze, and large geographical distance in the major malaria hotspot in north and western Brazil. As Dr. Alfred said about genomic approach, when we use genomic epidemiology approach, we can help to map source populations and prioritize areas for target control and interventions, and to eliminate residual vivax transmission, such as this place that I just talked to you, and similar endemic settings worldwide. Next slide, please. This work was published 3 weeks ago. And if you're more curious about the other results, you can just take a look at our paper. Next slide. I want to thank Marcelo Ferreira's labs, the current lab that I'm in. I'm PhD there, and Daniel Nifs's lab in Broad and Harvard School of Public Health, to all the work and collaboration until now. And I'm open for the questions and answers.
Thank you very much, T. So if there's any questions, please post them on the chat. Okay. So we have a question. What do you think could be acting as a barrier to recombination between sympatric strains?
Oh, this is a great question. It's not the first time people ask me that. We clearly don't know what's going on in that area, but we could think that even an area that it's like smaller as this one, we have like the mosquitoes that they have this, a lot of humans they can bite, so they don't need to go to other place to go and have their meals. So probably what you are seeing is that the mosquitoes are doing like subpopulations, and then as we know, the recombination occurs in the mosquitoes. So probably you're seeing this because the mosquitoes don't go into other places in the city because they have this, all humans who can bite. But we don't have like any kind of barriers, like geographic barriers that can separate it. So could be this thing.
I have a more general question. What are the main vector species in this region?
Main what? Sorry, I didn't.
Species, so the main mosquito vectors.
Oh, the vector. Oh, here we have Anopheles darlingi or Nisoricus darlingi is the main species in this area. It's not the same that we have in the coast of Brazil. It's another species, but it's the main species. And as we know, we have some publications for our collaborators that we have this substructure inside one population in Anopheles darlingi. Difficult name to say, darlingi, and then that's why we think that it's possible the barrier between them.
Okay. We have another question. Could there be a selective disadvantage to hybrid strains that contributes to this barrier?
I don't know. Could be like disadvantages, but I don't know because we don't have any work in this area. So.
Well, if there's, we have more questions. Do hypnozoids provide vivax with a path to longer term dissemination and potentially across greater distances as people disperse within and between areas?
Yes, could be. We don't have any work until now about hypnozoids in this area, but could be like a way of doing this spread and entering again in this machine and making the diversity high, and even this recombination and these clusters that we are seeing. Could be this, because the people inside the city, they go to the other city that is closer, the CS city, and that sometimes they spend like times in other locations too. So hypnozoids can make like a difference in this situation, yes.